We've talked about how different sets of enzyme reactions in cellular respiration will break down glucose in order to harvest the energy and use that to make ATP. But the question is, how does energy from the sun get captured in order to lock that energy into glucose molecules? So in Chapter 8, we'll investigate those reactions, which are part of photosynthesis. So first, let's define some eating schemes for various organisms. We have autotrophs, which are literally refers to self-feeding, meaning they can make their own food. A photoautotroph uses sunlight to make food. So this includes plants, algae, and cyanobacteria, as shown in photos A, B, and C. There are also chemoautotrophs, like some of the archaebacteria shown in E and D, and these will utilize inorganic chemicals like hydrogen sulfate, iron, and ammonia to make food. There are also a class of living organisms called heterotrophs. So heterotrophs cannot make their own food, and they rely on consuming the autotrophs to supply their energy needs. So this includes animals, fungi, and most bacteria. Now, the predator that eats the deer that's in the picture above gets a portion of that energy that was created during the photosynthesis reaction that occurred in the plants that the deer actually ate. So if we take a look, an overview of photosynthesis, we see that energy from the sun comes into the photosynthetic mechanism in the leaves of the plant, and carbon dioxide will be included to make sugars. Water is also a necessary reactant, and the result is sugars being produced and oxygen being released. So you can see that this is an opposite to the cellular respiration mechanism that we looked at before. So the materials that are utilized, the reactants in photosynthesis, include water that is absorbed through the roots into the plant, carbon dioxide, which is obtained from the air as a result of small little pores on the undersides of leaves. So if you turn a leaf over and look at it under a microscope, you can see these tiny little pores called stomata. A stoma is a singular, stomata would be plural, and it is made up, the stoma is actually the hole or the pore, whereas the sides of it are made up of guard cells. And oxygen becomes a waste product that will exit through that stomata. And the last required element is sunlight. So here is a YouTube video that you can take a look at that will go through the leaf structure. And we'll take a look at that also just briefly in the next slide. So the basic photosynthesis equation takes 6 molecules of carbon dioxide plus 6 molecules of water. We add in energy from sunlight. The energy is required because this is an endothermic reaction. We have to have energy put in. We're going to produce sugar molecules, and this is the formula for our glucose, and oxygen is going to be a product as well. So this formula should look very familiar because it is the reverse of cellular respiration. So the process of photosynthesis, just like cellular respiration, has many, many steps. It's complex. There's 2 basic pathways that are required to complete photosynthesis. The first is called the light reaction, and it's called this because it requires light. whereas the second is called the Calvin cycle, or the light independent or dark reactions. No light is necessary for the Calvin cycle, but the Calvin cycle won't continue if it doesn't get the materials it needs from the first section, which is the light reaction. So let's go back and take a look at the photosynthetic machinery. If we look inside of a leaf cell, you'll see many chloroplasts. Here's one chloroplast. It has a double membrane, and then inside the liquidy part is called the stroma. Don't get that confused with stoma, which are the pores. There are stacked sections, and these stacks are made of thylakoids. And there is a lumen inside the thylakoid. So there's a space inside each thylakoid, and a stack of them is called a grana. So let's take a look at the various reactions that occur. We know from before that there are light-dependent reactions, and then there are light-independent reactions, or the Calvin cycle. In the light reaction, it converts light energy, so we have light coming in, and makes ATP and NADPH. NADPH, which is different than NADH, which we saw in cellular respiration. There's that extra P in there, but it still serves as an electron carrier. This occurs in those thylakoid membranes. So in the membranes of each one of these little structures, thylakoid structures. And remember, a series of them is a granum. The Calvin cycle, on the other hand, so this is in one chloroplast. It occurs all in one chloroplast. This isn't a plant cell. The Calvin cycle uses the ATP and the NADPH made in the light-dependent reactions in order to fuel the Calvin cycle, which is going to take in carbon dioxide and put those carbon dioxide molecules together in order to make sugars. As a result, as the energy is taken from breaking apart the ATP and the NADPH, we're going to generate NADP plus the oxidized form plus ADP. That'll be shuttled back across to the thylakoids in order to be used again for the light-dependent reactions. So what is light exactly? Remember that light is energy from the sun. It's electromagnetic waves composed of photon particles. And these do travel in a wave formation. So we see this oscillating formation of an up and a down. And the difference from the top of one wave to the top of the next wave, the crest, is called a wavelength. Now light and longer wavelengths, crests further apart, has less energy than shorter wavelengths. For example, red light has longer wavelengths and has less energy than shorter violet light, which has more energy. So in the entire electromagnetic spectrum, there's only a short region that we can actually see. There is light energy that is in longer wavelengths than we can see and shorter wavelengths that we can see. But what we can actually see is in the visible spectrum, and this is also the range that plants use in order to photosynthesize. So here we see the red, which has longer wavelengths, than the violet, which has shorter. Violet has more energy, red has less energy. So the visible spectrum runs from 700 nanometers, which is in the red range, all the way up to 400 nanometers, which is in the violet range. Plants in their chloroplasts have pigments which can absorb specific wavelengths of light. And specifically, those pigments tend to absorb in the blue and the red ranges. So there are chlorophyll A and chlorophyll B. Now I've colored them green because they appear green, but they are actually absorbing red and blue light. So they reflect the green light, and that's why we see them as green. Beta carotene is a type of carotenoid, and that in the very dotted lines, the very dotted line here is going to absorb mostly in the green region, and it's going to reflect orangey, and that's why we see it as orange. And carrots have a lot of beta carotene, and so that's why they're orange. Here is a short YouTube video that can go through and talk to you about the light reaction, so take a look at that when you have a chance. But I'm going to go ahead and talk more about the thackyloid membranes and the light reaction and the Calvin reaction, which are occurring in them. So in the thackyloid membranes, we have two groupings of light collectors, which are called photosystems 2 and photosystems 1. And these are the structures which are going to absorb light. So this one is photosystem 2, and this one is photosystem 1. They are both going to absorb light through their chloroplasts. So they're going to absorb light, but they are going to utilize that energy to excite electrons in different ways. So keep in mind that we have both in the membrane of the thylakoid. Now, in between the two, we have an electron transport chain. So that's what's here. So electrons are going to be gathered up by both photosystem 2 and photosystem 1, and there's going to be a passing of these electrons through the entire electron transport chain, and we're going to eventually make NADPH using those electrons. There are two enzyme complexes. There's an NADP reductase and an ATP synthase. So this is the NADP reductase, which is going to make the NADPH from the electrons, and we are going to reduce NADP plus in order to make this. And then there's an ATP synthase. This is the same ATP synthase that we saw in cellular respiration. So as a result, we are going to pump hydrogen ions through our electron transport chain, just like we did in cellular respiration. We're going to have a high concentration of hydrogen ions, which when they flow through the ATP synthase are going to undergo a chemiosmosis in order to produce ATP. So the result of the light-dependent reactions is going to be a production of NADPH and ATP. So here's a closer up view of those structures. We see the photosystem 2 here, the photosystem 1. Photosystem 2 is going to gather up the light energy. It's going to cause a chlorophyll, these are the chlorophylls, to excite and release an electron. Some of those electrons are going to be used in order to convert water into oxygen and hydrogen ions, and that's going to be the source of our hydrogen ions. And then some of those electrons are going to be shuttled on through the electron transport chain, again pumping more hydrogen ions in, over to photosystem 1. Photosystem 1 is going to then, along with that light which is gathered and those electrons that are transported over, are going to excite this transporter, which will carry the electrons through the rest of the electron transporter chain in order to make NADPH. So photosystem 2 and photosystem 1 have a light harvesting complex. So that's these green sections. This is photosystem 2. That's the first one. Photosystem 1 is the second one. And I don't know why they put photosystem 2 first, but they each can gather light. We see them each gathering light. The pigments in the light harvesting complex, which is the green part, it says reaction center, but this is the light harvesting complex. The reaction center is the blue, is going to pass the electrons to two chlorophylls, special chlorophylls in the center, and those special chlorophylls are going to cause the electrons to go to the first electron acceptor, which will then shuttle it off to the rest of the electron transport chain. In the reaction center, that's this blue, that light is going to excite that electron to leave the chlorophyll A, and as I said, go to the first electron transport acceptor and transport that on to the electron transport chain. So this is a redox reaction, but it's driven by light. That lost electron needs to be replaced. So in photosystem 2, the electron comes from, so this is going to be giving up its electron, comes from splitting water, getting that electron, and producing oxygen and hydrogen ions. Those hydrogen ions can then be used for the ATP synthase to make ATP, and this is where the oxygen is going to come as a byproduct of photosynthesis. So here again we have the whole process, and just like the electron transport chain of cellular respiration, this hydrogen ion concentration gradient, which has been created by the electron transport chain, is going to be used to fuel ATP synthase in order to add electrons onto ATP and join up and make a high energy phosphate bond. Now the second part, this is still within the thylakoid. So this first part all took place still inside the chloroplast. This all took place inside the thylakoid. The second part, the Calvin cycle, takes place in the stroma, or the liquidy part of the chloroplast. So this whole thing is a chloroplast. The ATP and the NADPH that were made in the light-dependent reactions are going to be utilized in this Calvin cycle, where we're going to take carbon dioxide and we're going to join up carbon dioxide molecules into bigger and bigger segments in order to eventually get to a 6-carbon sugar. So there's 3 stages to the Calvin cycle. And the Calvin cycle is, of course, a circle, kind of like we saw with the Krebs cycle. And so we're going to take 3 molecules of carbon dioxide. We're going to utilize an enzyme, which is called ribulose bisphosphate carboxylase oxygenase. That's quite a mouthful, but this is the enzyme in peach here. That is going to join those 3 molecules together to form the first 3-carbon segment. We're then going to use the energy in ATP in order to process that, and the energy in the NADPH again to process, so that we can get enough molecules. So that's skipping ahead of myself. The first part was the fixation. That's this part here. The second part is the reduction, where we're going to use the ATP and the NADPH to add electrons in order to join up all those molecules and make glucose. The last step is that we need to regenerate the starting material, which was the ribulose bisphosphate, so that we have a cycle. So we need to generate that and we're going to use some ATP in order to get back to here so that we can start this whole process over again. So this shows us a YouTube video for photosynthesis, and after you have finished the slides, please take a look at this and watch this animation. That concludes Chapter 8.